Hey everyone, Connor Wander here with another episode of Straight from a Scientist. This one is very special because if you're watching on YouTube, you've noticed this is a full video episode. This is our first of its kind. If you aren't watching on YouTube, go ahead and check out the channel Straight from a Scientist on YouTube. Give it some love because we really haven't sent anyone there yet. If you are on those other channels, of course, you're going to be missing out a little bit. And we hope to have many more video episodes coming in the future. Now, this one was with Lee the Scientist, Lebaron Agostini, but Lee the Scientist on Instagram. You can check out his contact links in the show notes if you want. We talked about cancer therapies. We talked about the different strategies people use for cancer therapies and what is on the horizon for that. And then we actually got into a bit of, different types of cancers, how cancer works in general. And then also kind of went on some really great tangents about drug companies and the ethics and the politics and just kind of what's going on in the medical space, especially in the United States where people are paying just exorbitant costs for medications. And we try to explain a little bit of why that happens, but then also, discuss some of the problems in the system that allow it to function as it does. Lee, it was really fun talking to him, and he's a super hard worker. You can see that come out in the podcast. I do apologize for some of the audio quality that comes out in the beginning. For some reason, these online channels we've been using lately aren't really up to snuff, so I'm going to move again to just pure live in-house interviews. Everyone will be face-to-face in the future, and we're going to try and cut out some of that online interview stuff. Unfortunately, means that we won't be able to talk to people long distance for a little while, but until we figure out some of those crackling issues that you had will maybe hear in the episode, we're going to have to figure out some other way. But it does clear up about halfway through, and the conversation is just amazing all the way through. Could have talked to Lee for hours. We did talk for quite a bit after the episode that I wish I could have recorded that too. But yeah, I'm sure we'll have him on again. Maybe he'll come visit or we'll go visit him. Anyways, enjoy. Again, this is episode 51. And check it out on YouTube if you are just listening on Spotify, SoundCloud, or whatever.
Welcome everyone, Straight from the Scientist podcast here. This is actually our first video episode. I have with me Lebaron Agostini, who's at Jefferson University, right?
Yes, Jefferson.
Thanks, Lebaron. Also, Lee is basically his name. You can find him on Instagram at Lee_the_scientist. That link will be in the show notes. He reached out to us, said he was a fan of the show, and he has some really interesting work, and we're going to talk about that. So thanks, Lee, for coming on.
Awesome, man. I appreciate the opportunity to talk.
In a nutshell, could you tell us a little bit about your research? I know it's cancer related, and
right,
that's where it ends.
So we study pancreatic cancer in the lab. There's two major brands of pancreatic cancer. PDAC, which is pancreatic adenocarcinoma or adenoductal carcinoma, and then there's neuroendocrine tumors, which make up like 10% of all cases, not as deadly, not as aggressive, different treatment modalities. So essentially we're like a lab that focuses on this protein called HUR, which is like an RNA binding protein. That protein regulates other transcripts. So one of those targets is actually the protein that I study called PARG, P-A-R-G. And essentially I've collaborated with UK Cancer Research, which is like they have a drug discovery unit. They produce some small molecules. Essentially, I'm doing target validation and then looking at this concept called synthetic lethality with other proteins as a method to kill tumor cells.
Okay, nice.
So there's like a lot there.
Yeah,
there's a lot there.
So there's your elevator pitch. But for our listeners who don't really even know much about pancreatic cancer, now from some of my friends who are in the cancer field, I know pancreatic cancer is one of the more deadly cancers that are out there currently. It has a terrible survival rate. It's
the survival rate for pancreatic cancer right now is 9% or like 9.5%, and that's trial data. But the reality is probably less than that.
Yeah.
Because in a clinical trial setting, everything is controlled. There's no hiccups, and we're monitoring everything. It's probably in the real world, it's probably closer. It's probably closer to 5. And even the therapies that we have now are they you have 18 months max.
Yeah.
Like the best therapy that we have, the standard of care now, the median average is 15 and a half months.
Whew. Yeah, it's terrifying. But I know there's all of these different types of pancreatic cancer too. You mentioned the PDACs, right? So how are those different from regular cancers?
So it's really about the molecular subtyping, essentially. So I don't know how familiar. So like, let's take an example that's like very well known. Like breast cancer was like subtype. There's ER positive, PR positive, and that tells you what type of receptors are essentially driving the cancer.
And then how to treat it.
Right, exactly, right? So if it's ER positive, then we use an anti-estrogen. Mm-hmm. Pancreatic cancer doesn't really have that. The last decade has been looking at ways to try to molecularly subtype the cancer and then use selective therapies based on the subtyping to try to create like new therapeutic modalities. And that's just been pretty much unsuccessful. The standard of care is kind of like a kitchen sink approach. It really is. I mean, I could talk about it further, but yeah. So there are some subtypes as far as transcription and genetic approaches, but most people just get the same therapy because the clinical data doesn't really support treating those tumors differently right now.
Yeah. And as far as cancer as a whole, if we can step back even further, it's always nice to talk at a base level so the people who are coming in with very little scientific training can get it. What is cancer at a base level? I think of it as kind of just runaway growth. You can tell I do this as I'm talking. Right? So runaway growth in the body, but we want our cells to grow normally, right? So what is the difference of a cancer cell?
All right, so you got to think about it like this. Everything is operating in kind of this not necessarily unison, but in this kind of coordinated fashion. Two cells are next to each other. They understand, hey, I'm this type of cell and you're this type of cell. We need to work together to do whatever this function is, right? They're programmed in that way, and then there's like genetic regulation associated with that. So what happens in a tumor cell, or before that cell becomes a tumor, there's some genetic mutation. Those mutations shut down some of the intrinsic pathways that stop cells from turning into tumors. And once enough of those pathways have been altered or dysfunctionalized, then the cell's sole purpose is growth. It's not about helping for this bigger scheme or bigger functionality. Its sole purpose is growth. And so that's why there's a common, I wouldn't say something called like de-differentiations. Tumor cells tend to be de-differentiated, meaning that they're not more like the final type of cell. They're more closely related to like the stem cell of whatever that cell type is, because they have thrown away the functional aspect of the cell and more focused on the proliferation of that function. So essentially, I like to use this analogy for cancers in general. Like cancers is like the word automobile. There's Audis, there's Lexuses, there's Mercedes. There's things that make them uniquely similar, like they all have tires, they all have doors, but they're also very different in the sense where if you've been fixing Mercedes and then you open up the hood to, my Chevy Malibu, you may have like, I don't really understand these types of things. And cancers are very much like that because they come from different cells, and treatments are very much like that.
But at the core of things, it's a cell that is not only no longer useful, but it's taking up resources of the cells that still are useful, right? So that's one problem. And then it can also actually damage the surrounding environment, right?
Right. So part of that kind of like that communication between two cell types when they're trying to do some function, once that mission has been canceled, where I need to work together with you and my sole purpose is growth, I don't care that you're next to me. Like I'm going to just grow as much as possible. And then it's about energy consumption and growth, all right? And it's kind of similar, if you think about just normal cells, they need macromolecules to get bigger and to divide. And so that becomes a primary function. everything about the cancer cell is associated with growing and survival, in a sense where they also invade the immune system.
Yeah, that's terrifying.
Yeah, kind of.
It's terrifying, absolutely. So I've gotten really interested in like what causes cancer. Not lately, I've always been interested in it, but just yesterday, actually, I was at this seminar, and the Damania lab at UNC came to present to the pharmacology department, and Dr. Damania said, hey, we've been studying this virus. It's a sarcoma-associated herpes virus that essentially hijacks cellular machinery and causes cancer. So I had known there were viruses that were associated with cancer risk, but I never really knew the mechanism to that extent. It's crazy. It's like it's injecting this basically will to become cancerous into cells, if I were to paraphrase it horribly.
Well, yeah. So it turns out that the virus's purpose isn't necessarily to turn the cell into a tumor. It's kind of like it's just by happenstance. What happens is some viruses are like replication dependent, meaning like they want the cell, that the host cell, to replicate. And so a good way to do that is to kind of change some of the cellular pathways so that the cell that you're infecting is replicating. Turns out if you do that as well, though, you increase the chances of a cell turning into a tumor. So it's kind of, and there's lists of this. Hepatitis B virus can cause liver cancer. HPV can cause a whole list of cancers. Kaposi sarcoma is associated with HIV infection. So that's kind of an interesting thing. That actually makes up like the minority cases of why tumors aren't. Most tumors aren't associated with viruses. Most tumors are associated with randomness. Like,
I've heard that you get technically kind of cancerous or precancerous cells all the time. You get mutations. People always talk about mutations in cancer. Basically, the code that normally regulates the cell breaks down, and the rules are changed for that cell, and that cell can now grow and grow and grow to its heart's content. Now, we get, I don't know the exact number, but we get a lot of those cells happening every day where their DNA gets damaged, but they're typically properly destroyed. Now, you mentioned immune evasion. It's the immune system that's supposed to handle that, right?
Correct. So, for example, you need sunlight to turn vitamin D precursors into vitamin D, right? Sunlight's good. But also it's the leading cause of skin cancer. So why is that? It's because UVB, which is like the energy that comes from the sunlight, creates something called a thymine. It's called a pyrimidine dimer. Essentially, you take two DNA nucleotides and you create a bridge between the two. In a covalent modification, right? You turn those two atoms to stick to each other. Turns out when the cell goes to divide that stretch of DNA, it has a trouble with that, and then that can create mutations. And so once you introduce those mutations, the cell can turn into a tumor. And so that happens all the time. And an immune cell, because the cell is being transformed, as we would say, it produces these types of molecular markers that lets the immune system know, hey, I'm stressed, and I'm also presenting proteins that I'm not supposed to be, I've never made before, right? Because if you think about letters in a sentence, or more specifically, letters of a word create the meaning, and once you change those letters, you get a different word. You get a different word, you change the sentence. And so the immune system picks up on those changes and goes, hey, I need to kill you. And normally that happens. And so there's a threshold in which tumor cells figure out how to evade the immune system. And that's usually when they get to a point where they're detectable. Your immune system is already taking a shit. It's losing that battle. Once you get to a physician, they say, hey, you have a tumor.
And is it not the case that in people who are immune compromised, their immune system isn't working properly, they have higher risk?
They do. They have a higher lifetime risk. For example, transplant patients have a higher lifetime risk for cancer because
they're
we have to
immunosuppressant drugs. Yeah. So, I mean, it's really interesting. And people always ask me about my field, which is Alzheimer's disease, like we were talking about, and there are all these risk factors for Alzheimer's disease. None of them are guarantees, right? So it sounds like the same thing is happening in the field of cancer. Not only are there a million types of cancer, but
right. And so there's different For example, there's inherited cancers. Your family can have a genetic mutation or an inherent version of a gene that predisposes you, essentially makes you more susceptible to whatever type of cancer. That's pretty common. It's at some probability. And so breast cancer is like one of these examples, breast and ovarian cancer with BRCA1 and BRCA2 mutations. If you have that cancer, you're much, much more likely to get breast cancer or ovarian cancer if you have that mutation. And I can get into the specifics as to why that is. But yeah, essentially it's a probability thing. There's risk factors, then that's the genetic side. And then there's also things you can do that increase your risk of cancer, like smoking and drinking alcohol.
Oops. I'm drinking a beer right now. I'm only going to have one tonight. I mean,
alcoholism is definitely associated. Well, let me just, not to name like specific things because people will get caught up on that, but poor lifestyle habits in general will increase your risk for cancer.
Mm-hmm. Yeah.
So not exercising, these types of things.
I do make sure to exercise. I always tell people I have like a 3-day rule. Even if I have so much work to do in lab, I do not mind taking some time off for that. And we can't talk about it enough, right? Like there is all of this scientific basis towards lifestyle, proper diet. And I'm sure that guides your life too as you stare at this stuff through a microscope or what have you every day.
Yeah, I mean, to be honest, it's one of these things. I think the public perception of cancer is a little different from cancer is really a disease of old age. Mm-hmm. The median age of onset is like 73. Old people get cancer. Their immune system is failing. They've had a lifetime of doing bad things, mm-hmm. and it catches up. Young people normally don't get cancer. If you're young, 30s, 40s, and you get cancer, then likely you have some genetic predisposition, or you've been exposed to some environmental onslaught that's put you in a situation like smoking, for example, will mm-hmm. dramatically decrease your age for cancer onset and also cancer risk because you shouldn't be inhaling smoke on a regular basis. It's kind of like, hey, why would I do that?
We're not built for that.
Yeah, exactly.
Fair enough. So I know your specialty is pancreatic cancer. Are there any environmental triggers for pancreatic cancer specifically?
Smoking actually is one of the only things that's associated with pancreatic cancer. We don't understand why.
Yeah, you wouldn't think of it. I mean, maybe it's just because it can suppress the immune system too, right? So.
Well, if that's the case, then because smoking isn't associated with all types of cancers.
Oh.
Yeah.
I would think that. Yeah.
It's not correlative with urinary tract cancer, but or foot, testicular cancer. It's not correlated with every type of cancer. So the epi data will tell you what things are correlated, but the mechanism isn't really well defined. We don't really know. And even pancreatic cancer is relatively rare. It's like the 11th most frequent disease. Only 3% of people get pancreatic cancer. Of all cancer patients, only 3% of them get that. Mm-hmm. The problem is 91% of them die within 5 years.
Yeah.
Once you get it. And if you look at, there's 50,000 cases a year, 55. There's 54,000 deaths a year. So, yeah. So that's really, that's the bad part. But it's not super. Currently, it's the third leading cause of cancer-related death. First is lung, and then it's colon, and then pancreas is third.
And are those just because lung and colon are more common generally?
True, but it's also because they're just more aggressive. I wouldn't say more aggressive because the aggressiveness is kind of a snafu kind of word. It means a lot of different things in different oncology contexts. But lung cancer is super deadly because of just the biology associated with it. There was recent breakthroughs in lung cancer therapeutics in the last 10 years that have really made a difference, but it's still the number one killer of cancer-related death. And colon is the same way too. We do colonoscopies and that dramatically increase cancer survival because we can catch it earlier. And you got a pretty good chance of surviving if you catch it early. But once it's metastatic disease, it just doesn't respond to therapies or you relapse. So there's just those types of issues.
Yeah, too little, too late.
Right.
So you mentioned metastases. So when a tumor metastasizes, it's spreading throughout the body, right?
Correct. Yeah.
So there's, I guess, a couple phases that I'm familiar with for cancer. There's the initiation, like the cell becoming cancerous. There's, I guess, promotion, which is the tumor growing, and then progression being metastases. So it's spreading throughout the body. Is that kind of a
I mean, yeah. I mean, these terms are kind of fluid, but yeah, you have a tumor initiation. A normal cell goes, you know what? Fuck this. I'm gonna turn into a cancer cell.
I'm out.
yeah. Like, this whole being part of the hand thing, I'm not with it. And then there's this initial growth, and then eventually, because of the kind of the conditions and the programming of that tumor cell, it'll spread to other parts of the body, and that's really what kills patients. It's the spreading to other parts of the body. If it's,
oh, sorry, go ahead.
I was saying if it's still the primary tumor, we could surgically resect that, and then everybody wins. Take the tumor out, and you're good to go.
Yeah. So what are the frontline treatments these days? You typically hear about surgery, so take the bad cells out, and then chemotherapies, which are stop the rest of the bad cells from continuing to grow and spread. What else are we using? Or
so I'll give you the I feel like every question you ask is like, oh, it depends, because we're really talking about.
Yeah, that's science, though, right? There's so much nuance, and that's why we have to have these long form conversations instead of like Twitter sound bites or something.
Right. Because we're talking about hundreds of diseases.
Yeah.
So you kind of hit on some of the key points here. Surgeries for early detection. Essentially, most tumors you can operate on, except for leukemias and lymphomas, because they're blood cancers. Brain tumors are really hard. Sometimes you just can't operate on them, no matter how big or small they are. They're just in bad spots.
Yep.
And then even surgery has two kind of flavors, where it's neoadjuvant, which means, hey, your tumor is in a place that's maybe kind of dangerous, but if we could shrink it a little bit, we might be able to operate on it. So we'll give a patient chemo up front, and then we'll shrink the tumor down. It's not spreading it, not metastasized. We'll shrink it down so we can operate on it. And then there's adjuvant therapy, which is we'll remove the tumor, the bulk tumor, but there might be some local spreading, lymph node involvement, but not metastatic disease. And we'll give you chemotherapy to kill maybe some of these smaller populations of cells that may have spread to the local area. So that's kind of like the first two stages of cancer, stage 1, stage 2, and then there's grades, which are about tumor size and things of that sort, but that's like more clinical. If you have metastatic disease, that means the tumor has left this organ of, entry, its primary organ. It's in the bone, or it's, for pancreas, things spread to the liver and then the stomach. Kind of just the whole GI area, bile duct. So if it's left the pancreas, then it's like, okay, shotgun approach. Here's a cocktail of chemotherapeutics, and we'll hope to kill it. But really, that's what we call like palliative care, meaning we're just trying to extend your life and make you comfortable, because once it's metastatic disease, we really don't have a curative option currently. And that's the case for a lot of cancers.
What are the kind of new strategies people are trying? I've heard so many like discussions of, okay, so we have these combination therapies, which I think some of which that's what basically you're working on, yeah?
Right. So I'm gonna break it into like 3 classes. There's I'll break it into 4 classes.
Sweet.
Right. So there's small molecules, which are drugs. Those kind of separate into 2 classes within themselves. Chemotherapeutics, which its sole purpose is to cause damage to the tumor cells. They're based on cancer biology. Tumor cells will upregulate certain proteins and they'll divide faster. So chemotherapeutics take advantage of that aspect and kill cells, but they also have side effects because blood cells and skin cells also divide fast. And then there's targeted therapy. You have some specific mutation. That protein is driving your tumor, your cancer. We'll make a small molecule specifically for that protein. And if we take that protein away or stop it from working, your tumor will get smaller. That's kind of been done since the beginning, since the 40s. New small molecules have arisen. They get better and worse. You know, it kind of just depends. Then there's biologics, which are antibodies. Mm. Antibodies bind to surface proteins. And then there's a subset of class of drugs called ADCs, which stands for antibody drug conjugates. Essentially, some of the most toxic substances on the planet that we used to give in like the 30s, and we realized, hey, these things are really toxic. We can't use them, but we found out if you attach it to an antibody, then you can direct it to the tumor, and then the tumor will essentially engulf the antibody because it binds to a receptor, and then the payload will kill the tumor cell. So that's kind of been, yeah, exactly.
Yeah, Trojan horse system. Yeah, that's really cool.
So that's kind of growing. There's been a few approvals in that in the last few years for those types of drugs, but the limitation there is always, you know, if I'm wearing I'm wearing like a burgundy tie, cool, I'm flagging myself. You can make an antibody against this. The problem is always how many other cells are wearing that burgundy tie that's not a tumor, right? Yeah. So there's that. And then I would say there's cell therapies, which is really crazy. And so I'll try to give it like a basic overview here. The immune cells that we talked about earlier, imagine if, you know, they're losing the battle. Just the armed forces of your body, and the tumor's growing and they suck, right? Their morale is gone and everything. What we do in cell therapies is something called CAR T cell therapies. CAR stands for chimeric antigen receptor. And essentially we take those cells out of your body, retrovirally infect them, right? We genetically engineer them to express new proteins that are specific for the tumor, and then we put them back in your body. And so now we've upgraded them, we turn them into super soldiers, and they go and search for the tumor and just, you know, do immune cell stuff, which is essentially radioactive toxic stuff all over cells. So the problem with that is when your immune system gets turned on really quickly, there's some downstream effects that can be really bad. And, you know, even those things can cause death as well. So
do you get autoimmune reactions in that case? Yes,
you can. So for melanoma, you can target like melanocytes, which is what the tumor cell is. And then patients will get like bleaching of the skin, like it'll just start killing all of the yeah, so it gets kind of crazy. Or there's something called cytokine storm, which essentially immune cells communicate via something called interleukins. They tell cells to do certain things, and inflammatory versus anti-inflammatory. Turns out if you piss off a bunch of immune cells at the same time, they can like stop your brain from working, or they can cause other tissues to die, right? So there's definitely this window where, to be honest, most of these therapies are about figuring out the window that we can operate in between killing the tumor cell and keeping the patient alive. Because unlike bacteria, which isn't you, we can give drugs that are selective for that. But the cancer cell is you for all intents and purposes. It's an altered version of you, but it is you. And so the therapies that we're trying to create have to be either very selective for that subtle difference, or we have to be able to navigate within the window that separates you and the tumor. So those are kind of the big themes right now.
Because it all works the same way, essentially. It's just some parts are working too well in a sense.
Right, yeah. So the gas pedal is down in the tumor cell, and you know it's going 110 and then you're going 25. But if you take the gas pedal away, then everything stops. And so that's bad, right? Because things need gas pedals.
I mean, you could kill cancer by you could kill cancer spread by stopping your blood from pumping around, right?
Right.
But then there's an issue there, So I know it's all a double-edged sword, and the same thing happens in my field as well, is we're looking at this biology that, you know, one, it's messy, so there's all these different types, and then two, there's no biological free lunch, as Tim Ferriss always says, a famous podcaster. There's no biological free lunch. So if you change one thing over here, it's probably changing another thing over here. Yeah. And you got to make sacrifices. Yeah.
That's always the trick with, and so my background before I started grad school was like in pharmacology. And the theme for clinical pharmacology is like it's like this saying, I don't know who said this, probably like the first pharmacologist ever. He was like, the right dose, the right drug at the right dose for the right patient.
Yep.
And what that means is, it's kind of this precision. I can talk about this too, because targeted therapies and precision medicine is like a buzzword right now. Mm-hmm. And cancer has specific meanings, but ultimately it's about figuring out, those very subtle details, and not killing patients. And it turns out it's really hard to not kill a patient with when you're trying to kill a tumor.
Yeah, I mean, you got to take out some cells, and there's going to be collateral damage for sure. So
always.
Yeah, I do remember that phrase from my pharmacology classes too, pharmacology student as well. But while the bulk of my classmates are cancer, that is not my background. I would not pretend to be an expert there. So I mean, yeah, we basically went over the long and short of it. As far as your research, like, what are you doing right now in the lab? What are you excited about, and what's around the corner for you?
All right, so what am I excited about? There's this protein called WEE1. Have you ever heard of it?
WEE1?
Yeah.
No.
Okay, so this protein WEE1. All right, so first before I get into the WEE1 thing, let me define synthetic lethality. Mm. And so if you have a chair and it has 4 legs, the tumor says, I'm going to take one of these legs away. And that instability of the chair is actually preferential for me, the tumor cell. Hmm. What we try to do is say, okay, well, since you're missing that one chair leg, can we create drugs or therapies that take another leg of that chair away to collapse the chair? Hmm. So in that concept, from a genetic perspective, is called synthetic lethality. Essentially, another example of this is if you have 2 genes, A and B, you can take A or B away independently and the cell won't die. But if you take A and B away, then the cell will die. So the protein that I focus on, PARG, PRG, is a DNA repair protein. If you take it away, cells don't seem to give 2 shits about it. Like targeting it as a single agent, I've deleted it, so it's like, okay, whatever. But it turns out if you take that protein away, they're really sensitive to drugs, to chemotherapeutics, and also if you take other DNA repair proteins away, the cell dies. So this was a paper we just published over the summer. Essentially, we were saying to summarize the paper, it was if you target PARG in pancreatic cancer, you can make those cells more sensitive to chemotherapies. That was like the premise of the entire paper. And then what I'm working on now is kind of piggybacking off of that work. You have questions?
No, your mic is like really starting to crackle hard. I'm not sure what's up with that. Yeah, I was just wondering if it was a volume thing or something. Probably. You seem like you're getting like amped about this. I love it.
I'm moving to my, just asking me my research. I'm getting hyped.
Yeah. Getting ready in presentation mode.
Right. So I'll continue. So I'm piggybacking off of that project, and essentially this protein called WEE1 is a kinase, right? So it takes ATP and phosphorylates other proteins. And when there's DNA damage, the cell has different cell cycle checkpoints. There's G1 and S, G2, and then M, which is mitosis. So as the cell is dividing and damage occurs, it will check itself at these checkpoints. Well, in tumors, they delete those checkpoints, right? Because they want to proliferate. So for example, in pancreatic cancer, P53 is almost always deleted or mutated 95% of the time.
It's like a gatekeeper for the cell cycle, right?
Right.
So you can't go and make more of yourself right now.
Right. The example is like, if I were to like, this is actually a bad example, but I'm going to go ahead and say it. It was like if I like punch the kid in the face right before he goes on a date, and he's like, oh my God, I got a bloody nose. And then the P53 would be like, wait, you can't go on a date before you fix this, right? Because you're going to be embarrassed and like, oh, it's inappropriate. So go home and fix this. Okay, well, fix this, then you can go on a date. So you delete P53 and the kid goes on a bad date anyway, and bad things happen. You get a tumor.
Bleeds all over the dinner table. Yeah.
Right. That's a tumor now. So it turns out, though, before the cell can divide, there's other checkpoints. And so WEE1 is responsible for the G to the M checkpoint. And so it turns out there's this very fine balance between deleting some aspects of cell cycle arrest while maintaining genetic stability. Because you got to remember, all of the therapies cause DNA damage or attack the ability of cells to divide. Mm-hmm. So cancer cells have a different threshold for DNA damage. It's not that they're essentially immune to it, they're just the threshold is different. And so you can target that threshold. So it turns out if you delete PARG, P-A-R-G, and WEE1 at the same time, tumor cells really don't like that shit. They don't like it so much that they die. And so now I'm trying to figure out the mechanism behind that association, as well as model it in a bunch of different cell lines and cell types. And something else I do in the lab is looking at drug synergy, which is, if a drug does 50% and another drug does 50%, I can predict what the additive effect is. Mm-hmm. Turns out you can do that in such a way where you can predict what the additive effect is, and based on the real results, you can actually create calculations that dictate what synergy is. Synergy means 1 + 1 is greater than 2. And so I'm modeling that based on these kind of drug responses and these tumor cells. So that's what I'm doing. I'm really excited about this concept rather, because currently right now, that's kind of a big push in the industry, targeting DNA damage repair proteins and looking at synthetic lethality, because it's another way to look at a tailored approach. What mutations does a tumor have? Can I just delete another protein and then the tumor will die? But the normal patient isn't missing these two proteins, right? So if I just delete one of them, or if I make a drug against it, the patient should be otherwise okay. And so that's kind of like a really exciting thing. The problem is that concept has been around for a really long time, and there's now a push more towards it now. I guess there's always been kind of a push for it. But the BRCA1, BRCA2 thing with breast cancer patients, a drug came out called a PARP inhibitor, and that really brought synthetic lethality as a concept back into the light, because that drug has like revolutionized BRCA1 and BRCA2 treatment responses for patients.
So this WEE1, have you tried deleting it in like control cells? It doesn't mess with them. Like any normal cell can just be fine without it, or?
Yeah, because normal cells have P53.
Okay.
So if you delete the G2, so it turns out some cells will be susceptible to death if you delete this protein, yes. But there is some level of redundancy there, and normal cells aren't dividing at the same frequency.
Nice.
So, and this drug is already in clinical trials.
That's
so that's great, right? So people have kind of already figured, the tolerability out. Clinical trials fail for 2 reasons. They fail for a bunch of reasons, I'm sorry. But the most 2 frequent reasons are lack of efficacy, drug doesn't work, or safety, the patient's foot is like fell off or something like that. So they're in the stage where this drug will probably be safe, but will it produce a great enough effect to actually kill tumor cells? And that's kind of what's being investigated right now.
Yeah. Man, it's a lot of work. I can imagine you. And for the people who don't have lab experience, it's a lot of basically moving liquids around to different plastic dishes all the time, almost every day. and it seems like really abstract when you're looking at it from a top level, but to understand the biology, you actually really start to care about those, moving that one liquid from one dish to another. It doesn't look like much, I know, but so like, what were you doing today, if you can say anything about that? Like,
so today was, I know you know what a Western blot is, so I was, For those that don't know what a Western blot is, if you're just, listening in, you're like a, financial manager or something like that. A Western blot is a way to look at protein levels in a cell. Essentially, you take the cell, you lyse it, you burst it open, and then you look to see how much, how much protein is left in a cell compared to some condition. So currently, I was looking at a bunch of signaling pathways associated with WEE1 and PARG. Essentially, it was like, PARG, one control, WEE1, then a combination at different concentrations, seeing how that affects signaling. And so I'm kind of doing these experiments to prep for like large-scale proteomics, which is essentially like a big data approach to looking at changes at protein levels within a cell across like, different times or different conditions, things of that sort. So I was doing that today.
So basically testing if you start mucking with the WEE1 levels or the efficacy of that protein, what else might happen in the cell?
Well, yes, exactly.
Kind of predicting side effects more or less, or?
Well, I'm just trying to, in cancer biology and drug development, I would say, you want to de-risk the project, right? How do you know that your small molecule is killing because of the target?
Mm-hmm.
So the best thing to do is to take a genetic approach. If you have two drugs, you can delete either target or knock it down with like siRNA, which is like a genetic approach to reduce the levels of protein. And then you can see if you get the same effects. So the last 2 or 3 weeks, I've been doing genetic model of PARG, WEE1 drug, genetic model of WEE1, PARG drug, and vice versa. Then genetic, then knockouts with the small molecule in different cell lines. So I've been doing this in every like computational way possible, like every, order.
Mm-hmm.
And I've convinced myself, I'm like super skeptic. I've convinced myself that, okay, I think this is real. This effect that I'm seeing is real. So now it goes, okay, so why are the cells dying? I have a hypothesis. I think when the cells go to divide and they're missing these two proteins, their chromosomes fall apart. There's a science word for that, but this is essentially what happens. So now I have to go and test that hypothesis by doing some imaging that I have never done before and will probably be really cool and make some really cool pictures, but I need to figure out how to do it because some of the papers essentially take pictures of cells like every 5 minutes. Like there's a machine that does this because there's no way I can do this shit.
Yeah, I know. Automation is great, honestly, in the sciences. I can't get enough of it right now.
Yeah, for sure.
Automate everything I can. So, like, kind of a summary, you're taking all of these parts in the chain out one at a time and then replacing them and just, you're trying to fix an engine kind of, right? You're trying to like diagnose that engine, an Audi or whatever it is. I mean, in this case, pancreatic cancer pathways. You take one part out and you say, okay, if we take that out, we take 2 of these parts out, will it still run? If we switch them out interchangeably, will it still run?
Exactly. So, and then kind of seeing, what the downstream effects of that is.
Mm-hmm.
So
cool.
yeah, that's the vibe right now.
So you're a 4th year like me. Is graduation on the horizon for you?
I have a projected. I guess we all have projected timelines, but that doesn't mean anything.
Don't ask us about that, even though I just asked you about it.
I'm trying to graduate early 2021.
Nice.
So this is the start of my 4th year, probably the start of yours too.
Yep.
So this time next summer, hopefully I can have a manuscript together, submit it by early, like fall 2020, reviewers, comments come back, whatever the case may be, and then defend in January. That's of 2021. So I got about a year and some change, like a year and a half left or something like that.
Mm. And do you have a plan after that? I know before the show you were saying you have a lot of kind of business side hustles things going on.
Well, I wouldn't call it a side hustle because no one pays me for this stuff. So
you're laying the groundwork, right?
I'm laying the groundwork. So I want to get into some of the stuff I do at the university aside from science is we have this student organization called BizBio. This stands for Jefferson Business and Biotechnology Group. Mm-hmm. We essentially train PhDs and postdocs for industries outside of academia. Educate them on these other opportunities. Me specifically, I want to go into consulting. Essentially, would be like, hey, company X, we're a small pharmaceutical company or we're a small biotech company. We have an issue, we don't know how to solve it, or we want to de-risk our project or get to a clinical trial or whatever the case may be. They'll hire a consulting firm to look at the situation from an outside perspective and give them a recommendation. So I want to do that fresh out of college, essentially, for the second time. college.
Yeah.
And so I can get into venture capital because I really want to be the person that employs or deploys rather the funds to, you know, kind of invest in these ideas, and not for the sake of money, but for the sake of funding really good ideas,
instead
of bad ones, because I don't know if you've ever heard of this company called Theranos.
Yes.
There's a book. It's written by the author. The author's name is he's like an investigational journalist, but I forgot his name. It's called Bad Blood. It's the story of Theranos.
Yeah.
And just like a quick summary, this chick that was like the female Steve Jobs tricked a bunch of investors and mega companies like Walgreens into believing that she could test hundreds of blood tests from like a drop of blood. And this went on for like almost a decade. Turns out she can't do that. And everybody...
no one can do it.
Right. Every scientist would have known that up front, but she got money from venture capitalists and people that didn't know anything about science, right? And so like all that money was just lost in the idea that it was bad. And so that sucks. Turns out, you know, people may not experience, you know, that type of failure because like, it's not like I'm investing millions of dollars. No one I know is investing millions of dollars in companies, but institutions they invest in VCs and venture capital funds. And so that money goes to waste. It's bad for the economy. Every investment can't be a good investment, like statistically speaking. But I think if you understand the industry, whatever it is, Alzheimer's, cancer biology, and you understand the business and the finances of it, then you can guide that capital to end up in places where there are really good ideas and that can make an impact in people's lives.
Yeah, I mean, you're going to lower the risk at the very least. It's still going to be a bit of a crapshoot, but at the very least, you're going to be more targeted. So yeah, it's a really cool goal. So you have things figured out a lot more than I do right now.
Or you want to stay in academia?
I honestly don't know. I mean, there's things I love about academia. There's things I hate about academia. Same thing goes for industry. You know what I mean? And so really, part of what this podcast is having me talk to people, and like I get to ask you questions about, you know, what are you interested in? Maybe I could see myself doing some sort of consulting and stuff like that. So
yeah, I mean, you know, offline, people have hit me up like randomly on like social media. You know, my page has an okay kind of following. And so I post about consulting sometimes, and people are like, hey, you know, tell me more about this. I'm just like, okay, I'm going to send you an email that's going to have like 17 PDFs in it. Because I really, part of the reason I do this stuff on campus is, one, to, you know, get my own network and experience, but it's also because there's a lot of students that really love science, enjoy science, hate academia, don't really know what their options are, you know, don't know what their value is in the industries, essentially. And I think if you have a PhD, the university does, not all universities, my university does a good job of empowering students, but traditional academic settings don't do a great job at telling you, hey, your PhD is valuable, yes, because you're an expert in this field, but it's also valuable because you spent the last 4 years figuring out something that no one else would be willing to do because it's ridiculous. And that gives you a set of skills that are transferable in multiple industries. You know, PhDs are great writers because if you've ever written a scientific paper or a dissertation, for crying out loud, you can write anything.
Not there yet, but yeah.
Yeah, it's painful. I mean,
there are grants, you know what I mean? Like being able to articulate an idea, set a plan, and then sell that plan to someone. I mean, that's useful, useful, if not more useful in business as it is in academia. So
yeah. So yeah, I mean, it's really, I think it's important. I mean, ultimately, you need people to go back in academia and be professors, and I don't ever want to discourage anybody from doing that. But for the people that are kind of indifferent about their careers, you know, they're looking for something else, still sciencey. There's just a slew of careers for people with PhDs or people that are very interested in science to be on the business or administrative side of science, and still enjoy their work and still have an impact. I feel like a lot of people do this because we want to have, you know, an impact. We're researching diseases so we can make a difference. And I think we look at business in this kind of way, like, you know, it's about profits. Yes, it is. Business gets a super bad rap, but ultimately, These therapies, these treatments, yes, companies make them, but if they didn't exist and they weren't being funded by dollars, these patients would, it would in the 80s, pancreatic cancer's 5-year survival rate was like 5%. Mm. And so it doubled. It's still a horrific disease, and 9% isn't great compared to like breast cancer, which is like 48.5% or something like that. But these problems are really difficult, and you have to throw money at them because it just takes so much resources. And throwing money at a problem doesn't usually solve it, but putting smart people on it usually does.
Smart money, yeah, exactly.
Right.
Yeah, you can guide the money. I mean, yeah, a lot of people don't realize just how expensive science is, and I mean, everything you use, you can really only use it once if you're doing it right? You don't want to wash something and then have it be contaminated by the last drugs you used in that well. So it's really sad, but that's just the hard truth of it.
Right. Yeah, it's crazy. I mean, Tufts University came out with like a report or like a paper recently estimating the cost of drugs, and it was like close to $2 billion. And it's like, that doesn't make any sense. Like at face value, like how could it cost so much money to bring something to market? So you need it. But when you actually look at the cost of breakdown, it's like you have to incorporate hundreds of institutions, like globally, in the U.S. alone, hundreds of patients, doctors, nurses. There's so many moving parts over such a long period of time. And everybody involved is trying to make a living and pay for their family. On top of the reagents, the things that you're trying to do to manufacture are like inherently expensive. I think there's a whole industry that will emerge in trying to make clinical trials cheaper. And you kind of see that a little bit with like AI and machine learning and like something called real world data, which is like EHR data, like electronic health records, and kind of like clinical data from not clinical trials, but like looking at the population. Can we use some of that information and use analytics to like create better clinical trials? And so I think the future is going in that direction, because only like, I think it's like 5% of, or less maybe, it's like 1 in 20 drugs that are like lead compounds from pharmaceutical companies get approved. So 19 of 20 things fail, and it's like hundreds of millions of dollars, if not billions of dollars, are blown away because it's just such a difficult problem.
Well, it's also getting harder and harder in a sense because, one, we are physically running out of compounds in a sense, but two, you have to prove that your drug is not only safe, which is a big threshold, of course, but then better than the current standard, which is, of course, necessary too. We don't want to just be giving some like half-assed drug therapy.
So, I mean, you don't see that in the diseases that we focus on.
Sure, yeah.
You don't see that because it's like a recurring model. Whatever the therapy is, some percent of the population is going to relapse, and then that essentially is a new disease.
Right, because it becomes resistant, it adapts, just like bacteria, antibiotics.
Yeah, it's not the same tumor anymore, and so you got to do something else, something new. So if you look at the drug approvals, you'll see that the approvals start to get really specific. Breast cancer after surgical resection, after third-line platinum resistant, like they get really specific.
Yeah.
And then you have diseases where it's not this is an interesting thing. Like rare diseases in like the 80s and 90s were forgotten globally. It was like, but that's actually a huge financial driver of the market now, because turns out the technology is earlier this year, a company called Spark Therapeutics, which is like 10 miles away from my university. They made like a drug for blindness, like a gene therapy. It was the first approved gene therapy, but it's like $300,000 per eye. Yeah, it's crazy. But it's like, okay, well, these people are blind, and they've been blind since birth. Yeah. So like, and even some of these cancer therapies, like the CAR T-cell therapy I was talking about, that's also like super expensive. But essentially, if you respond to that treatment, it's curative. That's kind of the trend right now. So it's like, is that worth trying it out and then seeing, and I think, drug pricing and all that stuff is kind of, in the government right now, and they're talking about an expense. And I think people will figure it out. Like legislation they'll iron out the details and come to a compromise. But yeah, I mean, rare diseases are getting kind of this new attention, which is good for patients because people that have these rare diseases, like no one gave a shit about them. Cystic fibrosis, right? Yeah, it's like even like cystic fibrosis. There's some drugs come out recently to help that, and before it was like you had cystic fibrosis, like you died as a teenager.
Yeah.
So it's just crazy. I mean, the whole landscape is kind of it's a good thing, like you said, that new drugs have to be better than the previous one, because that's a patient-centered view of the drug development process. Like, if it's not better, why bother? We're not trying. Yeah, why bother if we're not trying to improve? And so I know you probably remember from like pharmacology, there's like 17 ACE inhibitors, lisinopril and quinapril. They're like these Me Too drugs that pharmaceutical companies pull out, and you're seeing less of that now. And I think it's a good thing. It's hard. It makes the thing harder, but I think it's a good thing for patients in the long run.
Yeah, because they don't want to hear like, is Pepsi okay? You know what I mean? In the clinic. So, but I mean, essentially, we're like pricing miracles at this point with some of those rare diseases that you're talking about. This CAR T therapies. We're like, how much is a miracle worth? One, like, are people able to pay for these miracle drugs? And then two, at some point, it is going to become cost prohibitive for these companies to develop. So, yeah, like you said, in the political landscape right now, there's people saying, how can we let drug companies get away with, charging this much? And absolutely, some of them are taking a little more off the top than they should. But at the same time, if some of these therapies are so expensive to develop and there's so few patients that would eventually pay for them, that in order for them to be viable at all and not die on the runway, they're going to have to be expensive. So, I mean, maybe there is a subsidization from the government at some point, but yeah, I don't know the path forward, honestly.
Yeah, so if you look at the global revenues, so if a drug gets approved in the US, and let's say it has a diversified revenue stream, right? Like X money comes from the US, X comes from Europe, X comes from New Zealand, wherever, Timbuktu, doesn't matter. 50% of the profits, though, like 50% of that money comes from the US. Because we're essentially bearing the cost because other countries get to negotiate drug price.
Yeah, and that's, I don't know why we can't.
And it's, so essentially it's crazy. Have you heard of NICE?
It sounds familiar, but yeah, you're going to have to.
It's like an acronym. I actually forget what it is, but you know how we have the FDA here? The EU has a version of that, but they also have this thing called NICE, or NICE. I'm not sure if I'm pronouncing it right, but I'm American, so shoot me.
Too bad.
They essentially say, okay, is your drug going to enter our market at a cost that we consider valuable for the population? So for whatever disease it is, we're going to say you can price it at this much. We think this is reasonable. And if you don't want to price it there, we just won't let you sell it in the country.
So you have to choose.
Right. So that's why you can like drive to Canada and get the same exact drug for cheaper, because the price negotiation is different. And so people look at that and it's like a huge issue. And I'm like, well, yes, that makes everyone feel a certain type of way. But most of the drugs are not manufactured in the US. They're produced in the US, like the inception of that idea, the companies are in the US.
Researched, yeah, discovered.
Yeah, the research here, like, yeah, because that gets manufactured in like India or something like that. but and so there's just like this tricky thing. I mean, people have mixed, because the whole process, we're talking about people's lives and healthcare and improving their lives. It's like, I think everybody's experience, I would say broadly, everyone has someone that has been sick in their family at some point. And if there's ever been an instance where some medicine had helped improve your family member's life, you're very grateful that exists. At the same time, it also turns your stomach when like the CEO of some pharmaceutical company got like a $5 million bonus and like the drug is super expensive. Yeah. It's like, then on the flip side, it's like, well, it costs $2 billion for us to attempt to work. And you're like, okay, well, maybe that doesn't make me want to throw up as much. Maybe I feel better about it. It's like this very slippery slope of like emotions when it comes to drug pricing in the country.
And then also, if you look at the R&D budgets of some companies versus their advertising budgets, you might throw up in some cases too.
Yeah, I'm glad you brought that up, because that's kind of a it's a weird one. There's only like 2 or 3 countries that allow you to advertise to patients.
Yeah.
We're one of them. I think New Zealand may be another. I misspoke. I mean, I don't know. I think it's New Zealand though.
Sorry, New Zealand. If that's false, we'll look it up later.
Yeah, well, we need like a Joe Rogan guy to like be.
I know. Jamie, pull that shit up.
But yeah, it's kind of it. And I have a friend that who shall not be named, so he doesn't get any hate mail.
Sorry, Voldemort.
But my friend works for an ad agency, like a marketing agency, and his client, they do only healthcare, and one of his clients is like a pharmaceutical company, and so they pay them to develop a marketing campaign. And then, you know, when you Google something and it's got like that ad is like the first thing, they pay for keywords to be there. And it's like, okay, why does that matter, right? Yeah, it's like, you know, they pay to pop up for that keyword, and businesses do this all the time, but it's why are you marketing to patients? And this is like such a huge controversial thing about being able to market to patients, because not, you know, humans are smart, people are smart, but no matter what it is that you do in your professional life, I doubt that you have studied cure disease of that therapy as well as physicians. So when you come into the doctor's office and you're like, I've been, you know, brainwashed by this marketing campaign, I want this drug. You know, the physician is in an interesting position where he has to be like, I don't care what you want, or he's going to have to like cut you some slack and maybe prescribe you something that you're adamant about. And it just creates this dynamic that I think is not in the patient's best interest, because it should be like, what's best for the patient? Now, if that drug is better, cool, awesome. If the patient saw the commercial and it's like, the woman in the commercial is so happy when she got put on this new antidepressant, and I'm like, that's not what you should be going to your doctor about.
That's not how any of this works. No, and it's also disgusting how the doctors get brainwashed too. Like, there's people who wine and dine doctors to get them to prescribe more of A drug versus B drug, even though the studies on both are, you know, basically the same. Right. So, I mean, yeah, not only are the patients going in and paying doctors to listen to them pitch to them for a drug they saw on TV, but then the doctors are also possibly pitching to the patients in some cases.
Right. Yeah. I mean, it's, you know, kickbacks were like pure financial kickbacks were outlawed in like the 70s, I think. Like, but it's like, hey, you know, you're our number 1 prescriber. We'll fly you to Switzerland to give a talk at this conference, and, you know, this and the third. And I think it's kind of naive of us to look at that as not a kickback.
Yeah.
You know, it's consulting, it's all this, you know, but this, we're minting words here when it comes to these types of things. You know, the drug rep buys your whole staff lunch. It's like, I mean, it's kind of like, it's kind of an interesting thing. I don't really know. All my medicine friends are going to be like, Lee, you threw us under the bus on your podcast.
No, it's not everyone. I do want to say that too. It's not everyone. It's not all bad. Like, not all drug companies are bad. Not all doctors are taking, you know, under the table conferences, trips, free trips. But there are enough of them that it's worth talking about and worth telling people about, because it is a problem that could be fixed, and it's kind of cancerous as a society. So,
and in the same token, I would say, you know, lobbying, I don't know, you know, people have mixed views on lobbying. I'm going to just put it out there that I think it's a bad idea. But I would say patient advocacy, which is like something else that scientists could do if they're like not interested in, you know, get your PhD and you don't want to do science anymore. You want to like be an advocate for patients, or like go and do like science policy stuff. You know, the AACR has like lobbyists. Like, they go to Capitol Hill and ask for more money for cancer research. And like the NIH, I think a budget got passed in the House and in the Senate for an increase.
Yep.
And I mean,
that's moves. Yeah.
Right. And it's like, okay, well, how does that get done? Do politicians magically go, science is important? No, these people are debating about whether or not climate change exists. Obviously, there's things going on in a political landscape that we have to, you know, operate within. It's complicated. Politics is more complicated than cancer, as far as I'm concerned.
I think you might be right about that. I don't know much about politics either. So, but yeah, we got a lot of things that are broke, and only a few people working on it. So maybe we need PhD level politicians. I don't know what that would.
Yeah, it's one of the girls I work with, one of my lab mates. She's probably like the sweetest person I've ever met. Like one of the sweetest people. Like just like such a good heart. Like gets like emotional when we like talk to patients and stuff like that. Like really believes in what she's doing, and she's like really interested in science policy because she's like about making changes at a grand level that impact scientists and funding and like all these things so that we can do more of the stuff that we do, which is about people. It's kind of like hard. It's not to get like disheartened with kind of all of the things that go on, but I definitely think that we need more scientists getting out of the lab and like voicing their opinion, kind of taking part in things outside of just research so that there's increased visibility for people. I mean, like growing up, I didn't know a scientist personally. It wasn't like until like much later in life that I like even started meeting people that were like, yeah, my PhD is in like physics. I'm like, I'm a physicist. I'm like, that's not just like cartoon characters do that. Like, like yeah, I'm like a regular person. Like, you want to come play basketball? Like, physicists play basketball? Like, kind of like
they let you out.
Yeah. I think that's just really important for public perception as well as like, we're in a space where like people don't think science is important anymore. It seems. Like just the public discourse is looking at, and I think this is because we're in the age of internet and there's a lot of misinformation out there and a lot of people trying to pull fast ones.
And just because of academic culture at the moment is if you're not spending all your waking hours in lab, you're somehow like a screw up.
Yeah, you know what I mean? It's toxic, dude.
Yeah.
That could be a podcast within itself, just that topic about
work-life balance,
mental health in grad school. I mean, there was a Nature editorial recently about it. Actually, I'll send you. I have the PDF. I'll send it to you because it's kind of like one of these things where like academic institutions are like, well, we've always done it this way, and it's like, well, yeah, that doesn't make it okay.
Yeah, I know. We've done a lot of terrible things as human beings for a long, long time. So we're still reforming. So
yeah, people have to have like and I think it's funny because scientists are probably the most analytical and critical people you'll meet on a day-to-day, but it's also interesting to see how stubborn they are on, just humans at the end of the day. And it's like change is hard for everyone. And it's like, well, when I was in grad school, I slept here and I turned out this way. And I'm like, that's why you're an asshole.
I wasn't going to say it. I'm glad you did. Yeah. No, it's exactly true. Like we put blinders on and we say, okay, this is okay because someone else did. And with experimental protocols, oh, why did you do that way? Oh, because he did it that way in that paper.
Right. Yeah, it's crazy.
Creatures of habit. I mean, we're human, so that's what it is. At some instance. But yeah, I definitely want to have more in-depth talks about work-life balance. I'll plug PhD Balance, Susanna.
Susanna Harris.
Yeah,
she's awesome.
You heard of her? Yeah, she's just a machine. I don't know how she maintains a work-life balance, but she does.
She's on that no sleep diet. I know it well.
That's right. But yeah, it's so important and it's often overlooked, and it's I think one of the reasons why it's hard to get to know a scientist outside of the lab in the real world because we don't have that much time to spend in the real world.
Right.
So.
Which is kind of the upside of, social media gets a bad rap too. I think any technology, any platform, there's good and bad that's going to come with it. You can build things with the hammer and you can break things down with it. It's not the hammer's fault, right? And so this is kind of like this vibe where there's so much misinformation on social media, but social media is like how we are having this conversation.
Yep.
Right. It's like this. Exactly. It's like, different states, different countries, whatever, it doesn't matter. You can connect with people, and that ability to connect with strangers and be closer and share ideas and promote positivity is like one of the upsides to social media. And for scientists specifically, because we can't get out of the lab, it's like take advantage of these platforms to kind of, put your ideas out there and kind of bring light to some of like the issues that like, it's crazy. It's like being in the military. It's like, I wasn't in the military, but I'm just using it as an example. You know, an Army guy or a Marine guy will meet another Marine guy, and it's like instant, you know, camaraderie. And PhDs are like the same.
Yeah, fair enough.
It's like, you did a PhD, I did it. We've all been through like similar experiences because the way that the academic model is, it's like, it hasn't changed in like 12 generations. So like everybody kind of goes through the same things. And part of that is good because like it makes us like closer together via experience. But it's also like the graduate dropout rate is like 50% nationally.
Yeah.
Like, and we're not talking about people that aren't smart or don't have the grade. Like, these are students that get into graduate programs and are like, this PhD thing, I don't want to do this anymore. 50% across all disciplines. That's like, people should be thinking about, hey, you know what? Maybe we should change something up here. Like,
yeah, you bring in the cream of the crop and you're breaking half of them down.
Right.
So, I mean, there has to be an attrition rate, right? But I don't know if it should be that high. So, yeah.
Yeah.
Time to look inside and check some things out. Absolutely.
For sure.
Hey, man, this has been great. Anything you want to tell the audience, like maybe people who are now a little scared about doing a PhD, you know what I mean?
Right.
Like, I do want to preface, we all want, like, not all of us, but I think most of us kind of joined the PhD lines because we want to make a difference. We want to do something important. Some words of wisdom for maybe those college grads or
right.
about to be.
All right. So I'll give some brief like insight. I'll ask a question rather. Did you start your PhD right after undergrad?
I did not. No, I took 2 years as a technician. So I was on the vet track when I was an undergrad for quite a few years. And then it was like my junior year, I'm like working in a vet hospital, kind of getting bored of it. Not that the day-to-day wasn't awesome. I mean, I love animals, always will. But I just saw no progression. I could see myself as a vet doing the same things 20 years from now that the doctors are doing. And that seemed not so fun. So I wanted to like, progress. I wanted to do something new every year. I could do the same thing day to day, but for me, it was the constantly driving and trying completely new things that drew me to it. And then, of course, at making a difference. Like you can in a vet hospital too, but
but it's smaller scale. It's
exactly
animal by animal, patient by patient, so to speak.
Yeah.
So the reason I ask is because similarly, I was like pre-med, pre-pharmacy. Actually, I was like wanted to be a pharmacist because I've like always been into like pharmacology. And I worked at a pharmacy all through undergrad, and I was like, yo, I don't want to do this for 40 years.
Yep.
And I realized I don't want to dispense and talk about drugs to patients. I want to make drugs and understand therapies, right? And so that kind of led me through the PhD.
Nice.
But after I graduated, I worked at like the University of Miami at the Diabetes Research Institute for like a year, maybe more than that. Before I started grad school. And I think that year was just like really important to just be not a student before I started grad school. And the reason I bring this up is because grad school is just a very unique experience where you have to be mentally strong. You have to be professional. You have to be accountable in a way that you probably have never been accountable for, unless like you've had kind of unique experiences otherwise. But like if you've just been a student and, you haven't really been in the lab a lot, or like maybe a little bit of undergrad research is kind of really variable across the country, institution to institution. You might want to go and like get like a real job, be held accountable, understand like it's on you, because then you get to the graduate setting and then it's like, I don't care that you have classes.
Yeah.
Like you just gotta, I remember the first year in grad school, one of the exams I had was like, calculate the energy needed for these two proteins to separate.
Wow.
It was like, what was like the KD and like figure it out. And then I was like, do we have a small molecule approach? And I figured out the on and off rate. That was like one of the exams. And like everything is like essay. And it was like on top of studying for that insanity, it was like, we're still in the lab, like doing research, like essentially working more than a full-time job. Yep. On top of studying material that's like notoriously ridiculous. So unless you're used to that, you might want to be able to like prepare yourself mentally for, having amazing time management. I think like the things that you need to be successful in this setting is you got to figure out time management in a way that really works for you, not for anybody else. If you're like an early person, stick to that. Like don't, and if you're not an early person, don't join a lab where the expectation is for you to get in there at like 7. Like don't do that to yourself. I think people, one of the key things is don't join a lab for the science. Join a lab for the culture and the mentor. You're going to be there for 5 years, 6 years, maybe, who knows? The average of my school is 5.5 for my program.
Same for my department, actually, exactly.
So you're going to be there for, that you're going to be close with that person and those people. And the science is the science. You're going to learn science if you're in a good place, no matter what. But being your lab family really matters. And so be really weary about that, and talk to other grad students if you're thinking about going to an institution, because they're going to give you the 100. These are like all things I was like, because I actually had to switch labs in my PhD. I chose the science.
Yeah.
I was in a lab with a PI that just started, who shall not be named, and had just came from Vanderbilt, which is like, they have a top 10 medical school, and like, she came to the university with like 4 R01s.
That's a lot of money. Yeah.
That's crazy. So just for, the non-science listeners, our R01 is like the highest grant you can get from the NIH. Usually they're like $250,000 a year, or like they're like multi-million-dollar grants over like a 5-year time. She had 4, which is like unheard of for most researchers.
Yeah.
And a brilliant person, brilliant scientist, toxic, like the Wicked Witch of the West or something, like this horrible human being. And
She's working too hard, she doesn't have time to be nice.
I ignored all of the red flags, and I was like, there's great science here. I want to be trained like a great scientist. Turns out, bad idea. Not worth it. I end up switching labs and started in a new place with a great mentor. So just remember, don't choose the science project. Choose the mentor and the people in the lab, because you're going to spend like, I think the average grad student hours are like 60 to 70 hours a week. I think that's like what the average is probably. You're going to spend a lot of time in a lab. Yep. If you don't like the people that you're with, it's going to make it worse.
Headphones can only go so far. That's right.
Right. So I guess that's it really. Try not to eat like shit, because it's really easy to do that.
I meal prep every week. I got my lunches packed and ready to go every Sunday. So
see, you're a beast, man. I go through phases where like I'll do that. Actually, it's funny you said that, because I like to cook too, and I'll cook on a Sunday like for the entire week. And then there's weeks where like my pans will like collect dust on them because I haven't cooked for like 2 weeks, like not even microwaving, like nothing, like just eating out straight.
I don't know. I found that I burned more time going and finding the places to eat, unless you're constantly paying for delivery, at which point I don't know how you can do that as a grad student on that like it's too expensive. But it's just it's worth it to pay that upfront cost if you can. I understand if you're at work all day Sunday or whatever it is, you can't do it, but that's like my day.
So where are you at? What institution are you at?
UNC Chapel Hill.
Oh, okay.
Yeah.
Oh man, so you actually probably could like meet Susanna.
Susanna, we've met. Yeah, we've met multiple times. So I haven't had her on the show yet, but she's busy as hell, and
that could be like the first live like 2 people talking in the room kind of thing.
I've done that too, but I mean, it's just when I can, I want to talk to people from all over. So actually, you're in Philadelphia right now, right?
Yes, I'm in Philly.
So one of the guys we had on, I think it was episode 14, Ricky Ricksai on Instagram.
Yeah. We went to the same undergrad. Too.
Oh, no way.
Yeah.
Did you guys know each other there, or was it a big school?
No, he's a postdoc. It's a huge university. Like, huge university. He's older than I am. But he did his bachelor's at FAU. Then he did his PhD at UCF.
Yep, that's when I talked to him. He was finishing
Now he's
up.
at Penn. as opposed to, but we go to happy hour. It's funny you said that.
Nice, say hi to him for me.
for sure, man.
Yeah.
But yeah, I'm in the city, so food is kind of like, and Philly has like a great food scene, and like everything's close. It's not like you can just like leave the building and there's like,
that's true.
Jefferson University is like, it would be, I mean, it's literally in the middle of Philadelphia, like City Hall. Like I like hit it with a rock. It's like a few blocks away.
Nice.
So there's like so much stuff around, and that's like part of the problem because it's like there's so many options. I don't even.
Yeah. Not so much around here. Yeah, it's basically we have like a whole street of pizza shops, and that's pretty much it. It's not great. So you get sick of it pretty fast. No, there's a couple of great restaurants, but you kind of have to go and sit down for them to be worth it.
Right. You can't just run in there and get it to go.
Yeah, I'll have to come up. Maybe we can do a live episode with you and Ricky at some point. That'd be fun because he's in the cancer field now too. Yeah, so I've been catching up with him.
He's an immune guy, so he's on the he's working on a tumor microenvironment, which it's funny that I'm not a fan of this. I didn't even mention this, which is super important in the field, but I'm not a fan of targeting the tumor microenvironment. I think it's too complex.
I smell a debate coming between you two. I'll moderate it.
Yeah.
You guys go at it.
I think it's one of these things where I think in some cancers it's probably going to be maybe important, but there was a drug that came out called PH20 for pancreatic cancer, and it was supposed to increase the delivery of drugs to the tumor because the pancreatic tumor is like really dense and what's called desmoplastic, meaning like there's a bunch of immune cells that aren't working. There's a bunch of like fibroblasts and like all of this stuff. And like the tumor makes up a small percent of that. And so there's like interstitial pressure and that decreases the ability to drug. There's like all these complex things. This drug was supposed to target something called like hydrolonic acid. And that was supposed to break it down. Turns out in the clinical trial with standard of care, patients did worse. But all of the preclinical data was like greenlit, like, oh, this is great, this is going to change everything. Yeah, and then patients did like had not only did worse, like more side effects, but like their overall survival was less, and the trial had to be stopped. Like after everything, they were like halfway through the analysis. So, and there's no drugs on the market currently that affect the tumor microenvironment. I think messing with the immune system is good, but like dealing with like fibroblasts and tissues in the immediate area, I'm not sure that that's going to be
the angle.
Yeah, I don't think that's going to be the vibe.
Interesting. Yeah, it's the first time hearing of that.
He's going to look
that problem with that.
He's going to listen. Well, because it's really like a very pancreatic specific. So if you're not in this field, you probably wouldn't have like been privy to it. Like, oh wow, that didn't work. That's a problem. But because this is not a new idea, like the tumor microenvironment, we know it plays a role in like cancer biology. And so similar to Alzheimer's, that the amyloid stuff plays a role in the disease.
Right.
Is it the target to make the disease better though? Turns out the clinical data, no matter what happened, because I know there's mouse studies where if you delete amyloid, they get better.
Yeah. We've cured Mouseheimer's thousands of times. So
Mouseheimer.
It's the sad truth, man. And I'm still, we still use mouse models. There's nothing else right now. So
I mean, we do. We're in the same boat. The GEM models, the genetically engineered mouse models, are the best thing you can use for a disease if you can create a model that's like similar to the disease. But just it's an interesting thing. Like mice don't get pancreatic cancer normally. Mice get different types of cancer,
just not pancreatic. Huh?
But they don't get pancreatic cancer. Like you let a mouse live, it just won't get it. So we engineer them to get it, and then the disease looks like the human disease, but just because it works in that model. And part of the reason why drugs fail and like why it's so difficult to make therapies is because we really don't have a great model aside from what we've learned from other cancers to try to mimic, those types of models that really gives us enough predictive knowledge. And that example with the PEG-PH20 thing, again, it had great preclinical data, like, wow. And then it got into trial, and like everybody was surprised. Like the entire pancreatic field was like, oh shit, back to the drawing board. And I feel like Alzheimer's were probably the same way, like, oh, this thing is going to change everything. And then, oh, no cognitive improvement. Yeah, guess what? We're wrong.
Yeah, the anti-amyloid therapies, we were chatting a bit before the show about this, not looking great. And I'm kind of the anti-amyloid, I'm kind of in the camp that anti-amyloid therapies probably aren't going to be that successful, just because of, like, we should probably figure out exactly what amyloid is doing first.
That's still unknown. Like, you don't know.
So yeah, some people think that it's basically an ancient antibody system, which I think the evidence is strong for that case. We had Maya on recently to talk about that. And if you want to get rid of an ancient antibody system, it makes sense that patients are going to have immune complications, which some of them do in some of these trials. And then also it's part of a greater protein structure that if you get rid of that greater protein, you have just a whole host of cell cycle problems. So you need at least the parent protein to it, and then when that gets cleaved, that's when it causes damage.
Right.
So yeah, I mean, more complicated than it seems on the surface. So anyone wondering why we haven't fixed it, it's not because we're just twiddling our thumbs and wasting time and money. It's just biology.
You've probably never heard this, but like I get this all the time, and it's like, why is the government hiding the cure for cancer and stuff like that?
Oh yeah, I know.
I used to get really not really upset about it, but I've changed the way I run my Instagram page. I kind of got we're like all over this conversation. I kind of started to gain followers through debunking like pseudoscience stuff, specifically in health and in science. Like, vaccines don't cause autism. Here's why. Like, autism is actually a genetic disease. Here's the data. I would summarize articles, and then like, GMOs are not bad for you. Like all these different things. Like, the government isn't hiring cancer drugs. It's just really difficult. Like, all of these things. And it used to be a hub for debates. Like, if you go back like years ago, there's like, it's just ridiculous.
Bunch of toxic comment threads.
Yeah, really. Like, this is when I also found out the internet is very racist. It's like a racist place. Because I grew up in Brooklyn, it's like, I don't do it. And now I'm in Philadelphia, like, I don't deal with racism. But on the internet, it's like, it's a whole new world.
Because it connects racist places to non-racist places.
Right. And it's like, yeah. So anyhow, different comment we have. But I changed that style of like posting controversial comment posts or whatever the case, into more like educational, like more general science, educational stuff, talking about things that are kind of specific to mine, expertise, kind of talking about the graduate experience, more like we had a science march in Philly, was huge, when I documented while I was there, news type stuff, because I realized that, yes, that stuff's important to like debunk these claims, but it's just like a hub for this negativity. And I realized too, I'll post something positive and get 100 likes, 100 or whatever. I could post like something that kind of like ridicules science illiteracy and like 500 likes, 1,000 likes, and it's just kind of like the vibe where like that type of feedback made me like look at the page and be like, cool, we all here, we love science and stuff, but like I don't want to be the gatekeeper for looking at people that don't understand science as idiots. Like, that's not what this is about. Like, not understanding science is, yes, it's a problem, but like, we don't need to like ridicule these people into accepting something.
Yeah.
They don't need to just accept it. They need to understand it. And we're not going to bash them into understanding it. But comments like, you're part of this, you're a shill.
Oh man. I haven't gotten that yet. But yeah, I can imagine in your field that's probably a lot more prevalent.
All the time, man. It's also, you want to go into energy and work for a company? You want to do that? Like, you're definitely definitely a shill.
Now, you're doing it for the right reasons. I respect that, man. Very cool.
Thanks.
Look, this has been awesome. I'd love to talk to you again. Hopefully I can come to Philly sometime. We can get you and Ricky to go at it over tumor microenvironment. Not really. I'm just setting things on fire right now.
Right.
But yeah, man, anything else you want to say to the audience? Any last thoughts for us?
I'm going to plug the IG page, Lead the Scientist.
For sure.
Also,
we'll in show notes.
Yeah. Also let's connect on LinkedIn. I have, a growing LinkedIn presence. It's a totally different vibe. It's more like professional, more, educational for professions, that type of vibe. I don't put that stuff on my social media account, on my Instagram account, but I'd love to connect with people on LinkedIn as well. And if you have just other questions about cancer in general, don't be afraid to reach out to me via LinkedIn or Lead the Scientist on Instagram. I'm always super happy to engage people about cancer biology and like drug development, stuff like that.
Excellent. Thanks, Lee. So that's Lebaron, Agostini, Lee, and then we will put his Instagram in the show notes. I got to get on LinkedIn and actually update my page too, so I will see you there at some point.
All right, man.
Good to talk to you, man. I appreciate it.
Of course.